On your notes from yesterday, you actually had your sheet that looked like this. Really quickly, what happens during DNA replication? DNA is copied. You should have that.
And it's changed to RNA.
DNA is copied. That's the only thing that happens during DNA replication. The subunits are your four bases, A, C, G, and T. You should have that. It happens in the nucleus. The end product is identical DNA. This is not the whole translation and transcription yet. This is just DNA replication. That's it.
So it's not your cell?
No, because again, the only thing this is talking about is DNA replication. This is only replicating DNA. It's not the protein synthesis portion of it. Throughout, I'm sorry, stop yelling out. Raise your hand. Over here, too loud. Transcription. In transcription, this is when the mRNA is now there. And the mRNA is creating a complementary strand of the DNA. Remember, that's your messenger RNA. So that means the subunits are going to be A, C, G, and now U, not T. It's when it's copying information down, but everything is not copied exactly. This also happens in the nucleus. The end product is an mRNA strand because now it has that copy. Translation. Translation is when mRNA changes to tRNA. And when I leave, you can go back to the website and get this information if you did not get it all, because I am going to move quickly. And the website is on the top of this page, on the front, or on the top. mRNA changes to tRNA. And this is also, and I'm going to move, if you don't have it, you have to go to the video. This is also when it codes the 3 bases for an amino acid, and your amino acid then codes for a protein. The subunits here are amino acids. This happens in the cytoplasm and the ribosomes. Your end product is protein because it's translation. So the end product is going to be the protein. All right. So for mutations has a lot to do with DNA, and I'm going to move quickly, so then I'm going to upload the video. So when I leave, if you miss something, that's when you're going to go ahead and get it. But mutation is a change in DNA that affects the genetic information. When you think of mutations, you probably do think of Teenage Mutant Ninja Turtles, or X-Men, or some type of two-headed snake, or the hawk, something that's very visible. But after today's lesson, you'll realize exactly what mutations are. But again, it is the change in the DNA that affect genetic information. This happens when your base pairs are changed by the number. So there may be more or less base pairs. It happens when they're changed by the type. So instead of it being AT base pair, it might be a CG base pair. Remember, your DNA must copy exactly. If it does not copy exactly, it's going to create a mutation. Or the order. Instead of it being ABCD, it might be ACED. There are 3 types of mutations. For example, if we read this sentence, it says the fat cat ate the rat. This is the original sentence. When it's copied, it may come out the fat ate the rat. What happened? Cat was removed. So in your DNA, it could happen when you have the original sequence of base pairs and a base pair is removed. This is called deletion. Let's say in the morning you start breakfast with a waffle, eggs, and milk. But on a particular morning, you may be Russian and you go with eggs and milk. You're going to notice it's a difference, but it's really not going to make big of a difference. A deletion mutation will create a difference, but not a big one. Another type of mutation, the fat cat ate the rat, the copy, the fat cat XLW ate the rat. What happened? Something was added. There is an instance where something is added to the base pair where it's an extra base pair, and this is called insertion. Going back to the example with your waffles, egg, and milk, Let's say now you have waffles, eggs, milk, and coffee. You're going to notice, hey, wait, something else is out here. There's a little bit too much going on right now. So you will notice the difference in this type of mutation. The fat cat ate the rat. The fat hat ate the rat. Something was changed. This was a substitution. It's a wrong base pair used. So therefore, it's called substitution or silent. It's the same amount of base pairs, but one of them is different. So instead of having waffle, egg, and milk, now you have yogurt, egg, and milk. You will notice the difference, but it won't make much of a big difference. And this, if there's a substitution mutation, you'll notice it, but it's not going to make a difference in your living. All right? Now, if the strand, remember DNA has to copy itself, so when it unzips apart, and one of the strands is TGAGCCATGA, the other strand should be ATCGGTACT. Why? Because it complements it. T only goes with A, G only goes with C. It complements it. So this is the strand 1 and 2, right? Now let's say on your notes it asks you, let's say if this strand had to be recopied, but it's copied like this. Take a moment and look at strand 1, which is on the left side, and strand 1 and 2 on the right side. When DNA was copied and during the mRNA transcription and translation process, what happened? Didn't get replaced. How many base pairs are on the left? Insertion. There are 10 base pairs on the left, over here, and there are 11 base pairs on the right. So therefore it was insertion. Something got inserted. All right, moving on. There are 2 ways in which DNA can become mutated. One way is it can be inherited. You can inherit a mutation from your parents. So from parent to child. Another way is it can be acquired. Throughout your life, you can pick up something or be exposed to something in your environment, or just be through exposure, and it creates a change in your DNA. So it could happen where there's just a mistake where your DNA was copied. Or there's some exposure in the environment which damages your DNA, and that's from a mutagen. I'll give you a second to get this information. We're going to see that in just a moment. Again, if you didn't get it, this will be uploaded and you'll get it then. Write your question down, Alex, and I'll get to it. A mutagen is anything that can change and that causes a change in DNA. You'll notice that mutagens are also carcinogens. Carcinogens cause, which is considered carcinogen causes what disease? Say it louder. Cancer. Is cancer a mutation? Yes, because you learn from the copying of your cells when you played that game that sometimes cells continue to copy themselves when not needed. That's cancer. Sometimes cells are damaged and the damaged cell continues to copy. That's cancer. So mutagens are anything that causes a change in DNA. Some mutagens would be asbestos. Asbestos is the stuff that's in insulation in old buildings, and it becomes harmful when it's airborne. This could cause a change in the DNA. Another example would be cigarette smoke. And we all know what cigarette smoke looks like. Another example would be X-rays, so radiation or from the X-ray machines. Although these things are necessary, they could cause a change in the DNA. Hi. UV light. This could be from tanning beds or too much exposure to the sun. Remember, the ozone layer is to protect that exposure from the sun, but if we continue to deplete the ozone, then we are exposing ourselves to too many UV rays. All right, the following sentence using mutation as a mutagen, I'm going to move right through this so we can continue to move on. Basically, mutagens are anything that cause a mutation. And I'm going to move right past this, and you can go back and look at it in just a moment, because I want to get through. Significance of mutations, which means what is the important about mutations? We see that it has something to do with DNA and DNA being copied incorrectly. But I don't want you to worry too much, because sometimes your body can correct the DNA repairs, most times actually. So, and the DNA copies itself, the DNA through a feedback mechanism will do a double check and recognize, hey, something's wrong, I need to fix that. So again, your body will go through and say, we need to fix this so it corrects the problem. Some mutations can be beneficial. So there are some mutants who are walking around, and we say beneficial, not meaning you can read people's mind, or you can fly through the sky, or you can freeze time, or things like that. But some beneficial mutations where there are people who are immune to the diseases like HIV and West Nile disease. They can't get it because their DNA, there's something within their DNA that doesn't allow them to get it. And of course, scientists are studying those individuals to see if they can extract something or develop something using their DNA to provide the immunity to the world. Some mutations are deleterious. Deleterious means harmful, which means that if you have this mutation, then, or if this change in your DNA, it is going to be harmful for you. An example would be sickle cell anemia. This would mean that there's not enough healthy red blood cells in the body to provide the oxygen flow. Remember, your red blood cells are shaped like the one on the left with that dip, which allows it to go capillary movement through your capillaries and carry the oxygen throughout your body. The sickle cell, red blood cell, is shaped a little differently, like a sickle, and therefore it will clot and it won't allow the oxygen to flow through the body healthy. Another harmful thing that happens to DNA, which causes a mutation, is Down syndrome. And Down syndrome is actually a partial or an extra copy of the chromosome number 21. And this would, individuals who have Down syndrome have developmental and learning delays. Some mutations, however, are harmless. Harmless mutations would be, again, it's a change in your DNA did not copy correctly. There are individuals who are walking around in the world with two different eye colors, one blue, one brown. Nothing's wrong with them. The DNA did not copy. Your eyes are supposed to be the exact same color. They see perfectly fine, so it's harmless. How many of you have birthmarks? Beauty marks. Okay, mutant, 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 mutant. Me, mutant, I have a birthmark. Birthmarks, remember, your body is supposed to look identical throughout. If you have a change in your body, the color is too dark or too light than the rest of your body, that is a change in the DNA because it didn't come out like the rest of your skin, and that's harmless. None of us are dying because of our birthmark. Some people are walking around with an extra finger. Nothing's wrong with them. This right here is a little bit more evident, but sometimes people have a little extra piece of skin near their pinky. That's an extra finger. DNA did not copy exactly, and they're perfectly fine. Well, maybe you have an extra finger? Oh, see, he's fine. He's actually very smart. All right. Only times that people only normally get it cut off is sometimes the parents want it cut off when they're younger because they think it's going to cause a problem, or the person just decides to get it cut off just for normalness, if you want to call it that. Albinism is another thing. How many of you know someone that's an albino? Albinism is just basically the lack of pigment. Albinism can happen in all organisms, humans, animals, and plants. People who are albino, there's nothing wrong with them. Just like I look different than you, they look different than you as well, and there's nothing wrong with them. But their DNA did not, something happened in the change in their DNA. So let's just talk about this really quickly and we'll end it. Most times when you see albino organisms, they have red or pinkish color eyes and their skin tone is pale. This would be an animal, so the hair is white. A person would have a lighter skin tone. Plants would have a pinkish or a whitish undertone of the plant. Now, we did not talk about yet Punnett squares, but we are going to talk about it after Christmas break, so I'm going to introduce it to you really quickly here. The capital letter is the dominant letter, the dominant trait. The lowercase letter is the recessive trait. Do not get that in your mind that if you have a recessive trait, you are inferior, or if someone has a recessive trait, they are inferior to the dominant person. What that means is the recessive trait is less prominent to show than the dominant trait. Got that? So, for a rat that's a normal pigment, like a brown rat, versus an albino rat, the combination of genes are going to be capital A, for it to have normal pigmentation, because there are two dominant traits. Now, a brown rat can also be capital A, lowercase a, because it has the dominant trait and the recessive trait, but the dominant trait is going to show. An albino organism, however, will have two recessive traits, because now there is no dominant trait there. Got that?
Yeah.
All right, in your box below where it says Punnett square, on the left box here, you're going to put a capital A. On the right box, you're going to put a capital A. That's the top, right and left, or left and right, sorry. And then on the left side of the box where you see the albino rat, the left top, you're going to put a lowercase a, and the left bottom, you're going to put a lowercase a. Now, what this is saying is if a rat that's brown with two dominant traits mates with a rat that's an albino, which obviously has two recessive traits, what are the percentages of their baby? What is the combination that their babies can be? So you put the Punnett square together, you come up with the combination. So you have the dominant A, just like when you're looking at a chart in math, the dominant A with the recessive A is going to give a dominant recessive trait, which the rat's going to be brown. What about this box? Capital A, lowercase a, because we have the lowercase a there. Same thing. In all percentages, all of their babies are going to be brown. So they all will take after the brown parent. All right? Same thing happens with people. It's a little bit more complicated, but it's generally the same thing. Now let's say if now we take a brown rat, which actually has a dominant trait and a dominant gene and a recessive gene, so you put the capital A in the top left corner or box, and you put the recessive A, the lowercase a, in the top right. And you're mating it with another brown dominant recessive gene pair, capital A in the top, the side left, and lowercase a in the bottom left. Their first possibility of a baby would be what? Or one possibility of a baby would be dominant, which would be a brown. What about to the right box? Right top, capital A, lowercase a, so it's still a brown baby. What about bottom left? Capital A, lowercase a, because you had a capital A at the top and a lowercase a down here at the bottom. What about this bottom right? This is where you have the albino baby, and the daddy will probably say, that baby ain't mine. But it is, because of the gene pair. So albinoism can occur if you have two parents who both have a recessive trait. You want to go ahead and put that. Two parents who both have a recessive trait. And we're going to end it here.